So it's my pleasure to introduce Rex Bernardo, faculty member, endowed chair in Maize Genetics and Breeding, I believe that's the name of his endowed chair. He's going to talk with us today about maize breeding and statistical genetics for his 5-year MAES project. So go ahead, Rex.
Good afternoon, everybody. Thank you for joining us today. It's a pleasure for me to talk a little bit about the research that I'll be doing in the next 5 years. And so, as we know, maize is a very important crop in the state of Minnesota. It's a source of food, fuel, it's a source of feed, and it's a product that's found in a host of different things that you can find in, say, a grocery store. It is a strong base for rural economies in Minnesota as well as across the nation. It's a bit of an interesting situation to be a professor of corn breeding at any university in the US, because unlike in, say, Jim Anderson's program, where Jim described beautifully how they breed wheat, in corn breeding, we don't really do corn. A professor of corn breeding doesn't really do corn breeding because most or nearly all, virtually all of the hybrids grown by US producers are produced by private companies. And so what I've done is instead focus my work on more basic and translational types of research that I hope would lead to new knowledge, would lead to increased information, would lead to better breeding methodologies that the practitioners in the private industry would then be able to implement in their own programs. The maize breeding program here at the University of Minnesota builds on a very strong foundation that was laid about 106 years ago in 1915 by H.K. Hayes, who was hired, among other things, to start a maize improvement program for the state of Minnesota. And in the last more than 106 years or so, there have been quite a few changes in the maize breeding and cultivation landscape. We've seen yields increase from less than 30 bushels per acre to now more than, say, 170 or 175 bushels per acre on average. And the types of hybrids have changed from double crosses to three-way crosses to single crosses. Biotechnology has become important. Genetic engineering has become important. Different tools are now being used by breeders. And yet, there are two things that remain the same throughout these years. Again, first, maize still is extremely important in the US economy. And secondly, maize has served as a model system for breeding for different crops. Many of the methodologies that are widely implemented across different species today, both hybrid species and self-pollinated species, have been tried and tested and developed in maize and adopted to other species. So it is in this vein that then I am proposing that I would do the research that I'll be describing here in the next several minutes or so. In the previous incarnations or versions of my Ag Experiment Station project, I've focused mainly on the use of what I might call as power tools in corn breeding. And this would include the use of single nucleotide polymorphisms or SNP markers. High throughput markers have become cheaper and cheaper. It will include the use of doubled haploids in maize breeding that would allow the development of homozygous lines in only 2 generations instead of the usual 6 to 7 generations of selfing. These power tools, so to speak, include year-round nurseries that allow maize breeders to now grow up to 7 generations of corn in 2 years. I might think that's even an underestimate now if we think that in many maize breeding programs, they even do embryo rescue to circumvent that seed maturation process and that germination process, and it speeds things up even more. And there have been power tools such as statistical methodologies and fast computers that allow breeders today to do types of analysis that could not be feasible a while back. So my previous project dealt with how to use these different power tools, but what I'd like to propose now in the current project in the next 5 years deals with 4 different emergent areas plus a 5th area that is not quite related to maize breeding and genetics, but at least has some relevance to the work that I do. So I don't have any slides, but I do have some visual aids. The first work that I'll be doing, the first focus area that I'll be doing in the next 5 years is a continuation of my work on targeted recombination. Targeted recombination refers to the ability to induce or to select for recombination points in the genome precisely where the breeder wants them to occur. The idea that if recombination occurs between these pair of markers, we're able to select a homologue, one of those two resulting homologues that would lead then to increased performance. The idea being that with genome-wide prediction these days, we can estimate the effect of each marker found along the genome. So for example, if we have a chromosome, let's say with 10 markers, we can estimate the effects of the alleles at each of those 10 markers. We can then, and let's say that those 10 markers are segregating between the two parents, we can walk, so to speak, along the chromosome and ask that question. If recombination were to occur between marker 1 and 2, and we get the product, how much yield would we get from that chromosome? If recombination were to occur between markers 2 and 3, how much yield would we get if we get the better homologue? And then we repeat that until we can then pinpoint where we think recombination ought to occur on that chromosome. We can do that for each chromosome. And the notion being, well, if we get that, then that's a little bit like a design, because we're not just relying on chromosomes, we're not relying on the recombination that we would get in a large sample of progeny, but we're actually trying to select for where those recombination points should occur. There are two ways of doing this. One is by CRISPR, theoretically by CRISPR. It has been demonstrated to work in yeast and in tomato. I'm not doing that. I'm relying more on natural recombination. The idea being that if we survey a large number of progeny on a given chromosome, their different progeny would show different recombination points, and then we can select for those progeny that carry a targeted recombination at that point that we're interested in. So in 2017, I published a paper that began to look at this concept of targeted recombination to see how much gain can we actually get if it becomes possible for us to either induce or select for recombinations in each of the 10 chromosomes. And my finding with the classic B73 Missouri 17 population is that if we can do targeted recombination, we would double the gains that we would have for important agronomic traits such as yield, moisture, test weight, and so forth. And so I don't get to say every day that there's a procedure that can potentially double yields, but that's what the data results told us, that yes, it is a potentially powerful method that would allow us to have substantial increases in the rate of genetic gain. That was with one population. Some students and postdocs working with me found that that if you look at now at several or many biparental crosses for elite maize germplasm, we get that same average of doubling of genetic gain. However, there is variation about that mean of doubling, meaning that in some crosses we expect more than 200%. In other crosses, we would see less than 200%. On average, you would get about a doubling of gain. We saw the same thing with different self-pollinated species using publicly available data on wheat, on soybean, on pea. And a postdoc found that the same exact result or same general result, that we have a doubling of genetic gains. Along these lines with targeted recombination, we also found that it is a potentially useful approach to introgress exotic germplasm. In a study we did, empirical study we did, not the actual crosses yet, but trying to see from empirical data how much gain can we get. We found that in soybean that it's a possible way to introgress exotic germplasm from plant introductions into elite soybean material. The idea is that instead of the usual approach of finding QTL, single QTL in exotic germplasm, the idea is to scan the exotic germplasm for larger chromosome segments. That if this larger chromosome segment is introduced into an elite line, would impart favorable characteristics for different traits of importance in soybean. Yield, maturity, protein, and oil were among the traits that we looked at. Aaron, Lorenz, and I are collaborating on this, and we hope to be able to do a proof of concept study to validate whether indeed we get the gains that we predicted we'd be getting. We also began with a postdoc to find ways how to efficiently funnel or to pyramid these or to stack these different target recombinations into the same line. So if you recall, what we're talking about is we have a targeted recombination on chromosome 1, could be on 3, could be on 5, could be on 7. But then what we want to do is to pyramid or to stack all of these targeted recombinations into a single line. And so with some modeling studies in Maize, a postdoc and I found that we can do that efficiently for up to 5 Maize chromosomes using what we call a multiple funnel method. And what, as the name multiple funnel indicates, basically you have different pipelines or funnels going on where you stack a pair and then you stack another pair and then you stack another pair. If you picture a March Madness bracket, if you would, where you have different teams in those different funnels, that's exactly the idea we have for a multiple funnel stacking for targeted recombination. Those are past results. Moving forward, what we'd like to do is to first to complete a validation study we have on targeted recombination. This is in collaboration with Bayer Crop Science. We're using germplasm and resources in collaboration with the scientists in Bayer, identified some early maturing germplasm, identified where targeted recombinations should occur. And this study includes 2 steps. One, we're developing a library of targeted recombinations, meaning that we're developing a library of lines that would have, for example, the targeted recombination on chromosome 1, but chromosomes 2 to 10, Maize has 10 chromosomes, are all the same genetic background. We do that same for as many chromosomes as possible. And by doing so, we then be able to estimate the effect of a targeted recombination on a single chromosome. At the same time, we're trying to stack all of these individual targeted recombination lines to have as many recombinations as possible into one homozygous line. Those experiments are ongoing. The yield trials are going to be held this summer at different locations in the Midwest. Other aspects of targeted recombination that we're planning to study include, one is better ways to estimate where these targeted recombinations should be. You know, for example, for those familiar with this process of genome-wide prediction, we're using ridge regression block, which uses a single shrinkage parameter for the entire genome. But we're thinking, can we come up with something better? For example, can we use a shrinkage parameter for chromosome 1, a shrinkage parameter for chromosome 2, and again, a chromosome-specific shrinkage parameter? Would that give us different results, better results perhaps on which chromosomes to target, as well as better estimates of the gain that we would get? And then finally, for targeted recombination, a last study that we're planning to do is trying to see what if we now begin to stack targeted recombinations, not from the same biparental cross, but from different biparental crosses. The idea being that in a breeding program, you have different breeding crosses that are going on at the same time. And the intent then is in each breeding cross to identify what you might call as a high value chromosome, that copy of a chromosome that carries a targeted recombination. Meaning that in cross number 1, you might say, oh, progeny number 100 carries a very favorable targeted recombination on chromosome 1. And then you could say, well, on cross number 5, progeny number 50 carries a very good targeted recombination on chromosome 8. And then the idea then is to stack from different sources these targeted recombinations. So in a way, it's similar to the concept of a multi-parental breeding population, except we're looking at specific chromosomes and targeted recombinations on each of these chromosomes. A second area of study that I plan to investigate is the use of sequence information in plant breeding. The cost of sequencing is decreasing these days. And more breeding companies have access not only to SNP information from, for example, SNP arrays, but actual whole genome sequence information. And the question has come about, how useful would the sequence information be for different traits that are quite complex, for which we don't even know the genes, such as grain yield in maize. And so for this work, I'll be partnering with a seed company. I'm still in the process of negotiating a postdoc and access to the resources and executing a contract. And so therefore I cannot name the company yet or share some of the details of this work. But some of the thoughts that have come to mind is, for example, with sequence information, will that allow us to gain better estimates of where targeted recombination ought to be? The way it is now, for example, if with the current marker density that we have, if we have a marker interval, any targeted recombination between this interval is treated as being equal because of the lack of any other information. Meaning if I have a marker on 10 centimorgan position and 20 centimorgan position, each of these positions, it's all treated as the same. But the idea is, would sequence information allow us to better pinpoint where targeted recombination should occur within this 10 centimorgan span, which, if you think about it, is actually quite a large chunk of the genome. A second thought is the more general question: will sequence information be any more useful than just the use of a lot of SNPs in a breeding program? More so in a species such as maize, in which we have doubled haploids, and the doubled haploids are typically produced with only one generation of meiosis. So entire chunks of big chunks of chromosomes are passed intact from parent to offspring. Sometimes we see whole chromosomes being passed intact from parent to offspring. So in previous work, we've found, for example, that with imputation, we can get by with genotyping with only 30 or 40 or 50 SNPs across the entire genome. And so from that perspective, maybe genome sequence information might not be helpful because of the redundancies that we would expect with a high linkage disequilibrium with doubled haploids. Nevertheless, the resources will be there to do a good study on that question of to what extent do we really need sequence information for breeding for different complex traits in plants. A third area of investigation that I would like to focus in is looking into the gains per unit time and the gains per unit cost. In the work that I've done, much of the work that I've done so far in the use of these power tools in corn breeding, it was more along the lines, can we use this tool? How much value will it add, without really putting too much thought on budgetary issues, as well as how does it change the time required to achieve this genetic gain. In talking to different breeders in the industry, there's a big emphasis right now on breeding pipelines. A breeding pipeline being a very specific scheme that is used for breeding the species by all the breeders for that species in that organization. And there's a lot of interest in trying to fine-tune or refine breeding pipelines to use these different tools, keeping in mind, of course, the costs that are involved, as well as the time from start to end. So I foresee myself doing more investigations on refining breeding schemes while keeping costs constant and while keeping budgets constant. One piece of work that I've done in this area already has actually has to do with genome-wide prediction. How can we use multiple cycles of genome-wide prediction in a breeding program within a biparental population? Interestingly, maize, the cost of breeding with one cross has remained constant over the years. It's roughly the cost of one subcompact car. It was the cost of one subcompact car back in the 1990s, and today it's about the same cost, cost of a subcompact car. So the idea being, okay, if the budget is the cost of a subcompact car and we want to maintain this time from start to finish, how can we optimize the breeding program to increase the genetic gains to the extent possible? One way is actually to substitute phenomic information for molecular markers. The idea being that instead of using SNP markers, we could use hyperspectral image data from unmanned aerial vehicles as a substitute or as a proxy for molecular marker information. There have been results that have shown that this method is effective. the predictive ability might be lower in some cases, but then the cost differential, if those images are cheaper to get, could make a difference in the end. Another aspect of this gain per unit time and cost that I'm interested in is this concept of outliers in plant breeding. If we look at genetic gains in plant breeding over the decades, they have occurred in two ways. One is through steady gains from steady and patient selection. But once in a while, a breeder would get really lucky and find a new cultivar that is a step change or has outlier type of performance. Speaking of outliers, an outlier I do think about is a basketball player named Sean Bradley. Sean Bradley was from BYU, and he played for, I think it was the Philadelphia 76ers who Sean Bradley played for. And the story goes that Sean Bradley was on a plane ride sometime and was found himself seated by a geneticist. And he and the geneticist got talking, and the geneticist was studying height in humans. And this geneticist said, "Can I have a sample of your DNA, because there's some analysis that I would like to do?" And Sean Bradley allowed his DNA to be taken. And his DNA was analyzed for different plant height variants or plant height genes that were known in the human database. And lo and behold, it was found that Sean Bradley did not carry any major genes for plant height, that Sean Bradley simply carried a very rare combination of human height genes that caused him to be very tall. I think he was 7 feet 6 inches. And if you look at Sean Bradley's height, he was 8.6 standard deviations from the mean. Remember that in a normal distribution, you can go about as high as 3 standard deviations. Sean Bradley was 8.6. And it got me thinking, in plants, are there different characteristics of the germplasm? Are there different characteristics of the parents we cross that might lead to a higher incidence or a higher probability of outliers? Given that they're outliers, it's actually hard to study, because by definition, an outlier defies the expectations from the model. But that being said, I'm interested in investigating what might be some factors, some characteristics of these parents that we cross that might lead to outlier type of performance. And this brings us then to this fourth area that I plan to study, and that is on trying to update quantitative genetics theory. So last year, I published a paper in the journal Genetics called Reinventing Quantitative Genetics for Plant Breeding. The main thought in this paper is that the foundations of quantitative genetics were invented back in the 1910s, certainly a very different era from what we have today. Back then, there was no way to trace the inheritance of genes from parents to offspring. And because there was no way to see which offspring got which genes from the parents, what quantitative geneticists had to do was resort to probabilistic models. Meaning that, of course, if a parent's a diploid, father has 2 genes at a locus, then there's a half probability that a child would get one of those genes. Along with that, there were assumptions to make things simpler of random mating, meaning that a key, for those who've taken quantitative genetics, a key assumption is that you have a random mating population. And why that was needed? Well, because with random mating, you can determine what the genotype frequencies are from the allele frequencies and vice versa. Now, the problem, though, is that in most plant breeding programs, you don't really have a random mating situation. You can have a random mating situation if you look at individual populations undergoing recurrent selection. But if you look at the entirety of breeding germplasm, it does not constitute a random mating population. Now, that being said, we now have ways to track the inheritance of genes from parents to offspring. And these ways obviously are molecular markers. So the impetus here is that we seem stuck in theory that was developed from a different time. And we now live in a different age where different tools and resources are available, but theory has not kept up with the tools and resources available. So what I'd like to do as part of my research in the next 5 years is dig a little bit more and try to look at that question. What parts of the theory do we need to now set aside, saying that okay, this part of the theory has outlived its usefulness, but now what other new theoretical developments are needed to accommodate these new tools that we currently have in plant breeding. Now, these four different aspects that I've mentioned, I would characterize as having very little, if any, social or cultural component. As many of you know, I'm now doing some work with a plant breeding center here at the University of Minnesota. And one piece of work that I got interested in is actually vegetable breeding. And you say, well, how does that relate to maize breeding? My argument to you is there's a vegetable called baby corn that's still corn, right? But kidding aside, I'm interested in doing some work on vegetable breeding with local immigrant communities. And this work will be done through the Plant Breeding Center here at the University of Minnesota. The context is that we here in the Twin Cities are blessed with a rich immigrant population. These immigrants would like to continue their way of life. Many of them were farmers in their homeland, and they'd like to continue farming here, and they also enjoy their cuisines from their homelands. A difficulty has been a limited access or a lack of access to seeds or improved varieties of some of these vegetables that are important to local immigrant communities. So with a working group that has been formed at the center, as well as with extension horticulturists and other professors in the Department of Horticultural Science, I'm helping coach and guide this working group in this effort, providing advice on germplasm acquisition, on field trials, helping people network, and so forth. And so it provides a bit of, again, cultural and societal context to this overall work that I'll be doing. Thank you for listening to me about my description of my research in the next 5 years. It's been a wonderful ride these last 21 years of my being here doing work in maize breeding and genetics. And I look forward to the next 5 years as to what it would bring, along with other opportunities that are yet unknown, that I'm unable to describe today because I don't know them yet. But if history is any guide, it turns out that by year 4 or year 5 of an MAES project, there are new ideas that come around that I'm unable to describe today, but I hope I can describe in the next iteration that will be close to 2026. Thanks so much.
Thanks, Rex. Are there any questions that anyone has for Rex? So I have a question. So with regard to the targeted recombination, do you have evidence? So there are regions of the genome that don't recombine very much. Are your population sizes large enough to access those particular regions? And if so, do they contribute to the regions that are actually desirable to have recombination in those regions?
Yeah, it's a great question, something we thought about. So the work we're doing focuses on natural targeted recombinations, just selecting recombinations where they already occur.
Yeah.
But I think that's a very fertile avenue of investigation. It's like looking at the far side of the moon that we did not see before. And so you may surmise, yes, there could be hidden recombination there that could be useful. But you could also argue, well, maybe there's no recombination there because recombination there is bad. But then you could have a third argument, well, we're improving the plant, not for the plant's benefit, but for human benefit. So maybe there's recombination bad for the plant, but good for human benefit. So we don't know, but I think that's an area, possible area of investigation, but it would require more biotechnology types of approaches rather than the natural recombination that we're looking into.
So with regard to the vegetable breeding, what vegetables are you planning to work on, and what local immigrant communities are you working with?
The vegetables, the target vegetables will be finalized. Well, it could be it'll be not finalized, but identified with help from local immigrant communities. So far, the list includes vegetables that I am not familiar with at all. They include jute mallow, spider wisp, Ethiopian cabbage, and some hibiscus species that we look forward to learning. And these are important to the Somali and Kenyan local immigrant communities.
So the genetics of those of those vegetables? Are they outcrossing species, or are they selfing species, or is it all over the board?
No, it's a great question, because when I learned about these species, guess what I did the next hour is I went on Google and tried to find out as much as I can. I'm pleased to report that they are well behaved, I think, genetically, that they are all selfing species for which hybrids are possible. You can make crosses quite easily. I think a cross in some of these one hand pollination can give you many seeds, which bodes well for seed production if it were a hybrid cultivar. So, I think the outlook is positive so far. Do any of these vegetable crops pose a potential risk from an invasive species concern? The answer is yes. So we will be focusing on only those species that do not have invasive weed concern. In fact, that already would be a concern even importing it to the US. We would have to ask for germplasm from other parts, from GRIN, of course, in the US, but from other parts of the world. And if it's flagged as an invasive species, we most likely won't even be able to import it, and we wouldn't want to. But yes, that is a concern.
So I have one other question. So maybe I was daydreaming at this part in your talk, but what is the parts of quantitative genetics theory that you think is really in specifically that you think is outdated and you might be able to actually update?
Okay. A key concept in quantitative genetics is what's called the covariance between relatives, meaning that relatives resemble each other. Uncle, uncle, niece, parent, offspring, they resemble each other. But that ranges from 0 to 1. But there's no such thing that has been developed yet called a negative covariance. It's not possible to say that, well, as the value of individual goes up, the value of the other individual goes down. So to my understanding, that hasn't been investigated yet. And it is possible that when you have structured kinds of populations, when you have non-random mating, structured subpopulations, that type of covariance or that type of association might actually exist. It's not just 0 to perfect positive. It can be perfect positive to something negative. So that's one area that I'm trying to look into.
So if there are no other questions, let's give Rex a hand. I really enjoyed that talk. That was great. So thanks a lot.
Thank you. Thanks. Bye, everybody.
Bye.